Assalamualaikum and a very good day. I'm Doctor. We are from Group 2 who are going to present on the topic of dark field microscopy. We have 5 people in our group and I am the representative of the Group 2 who is going to take part of the first part of the presentation while my partner Muhammad Adham will take part on the second part of the presentation. So shall we begin? Dark field microscopy is defined as a technique that uses the dark background to view brightly illuminated specimen. Or in another case, we can say that it is used to study the microorganisms such as algae, hair, fibers, and also insects. The general idea of this microscopy is that it provides a background with high degree of contrast and can make samples with difficult backgrounds to appear more distinct or stand out with using minimal effort. So to explain the mechanism on how does this microscope works, first it requires blocking most of the light that ordinarily passes through the specimen, which will then allow only oblique rays to interact with the specimen. Next, when a specimen is finally placed on the slide, the oblique rays interact with the specimen and are refracted by the specimen in the sample. This will allow only a faint rays to enter the objective lenses. All in all, it will finally result in a bright specimen on a black background.
Okay, so thank you NURIN for presenting her part. So my name is Muhammad Adham Bakri and my train number is 200737. So I would like to present about the most important component in dark field microscope. What is dark field microscope is a good imaging technique and the problem encountered and solution. Okay, so for the most important component in dark field microscope is a condenser. Almost any bright field laboratory microscope can be easily converted to perform a dark field illumination. This can be done by changing its condenser, which is this dark field condenser designed for the dark field illumination, as you can see in the figure 1. So this condenser are relatively simple and offer the high numerical aperture or DNA and required to create the cone of the illumination needed for dark field. So as you can see in figure 1, you can see the cone shape that creating the dark field illumination. And many condenser can accommodate insert that can create a cone illumination, but this insert don't offer an A as a height as a dedicated dark field condenser, and this so not all the objective lens can be used. Okay, so however, the insert of this condenser can give you the flexibility to have a multiple observation method on the same condenser. And for the wide dark field is a good imaging technique. Actually, in general, the object imaged under the proper condition of a dark field illumination are spectacular to see. So you can say that often specimens are containing very low inherent contrast in the bright field microscope shine brightly in the dark field microscope. Okay, so that's why you can use a dark field instead of a bright field microscope. And usually dark field illumination is the best for revealing the outline, the edges, the boundaries, refractive index gradient. So these all candidate for dark field illumination also include minute living aquatic organism, diatom, small insects, bone fiber, hair or unstained microorganism, yeast, and also a cell in a tissue culture. So usually, but non-biological specimen include mineral and chemical crystal, colloidal particle, dust crown specimen, and a thin section of polymer also can be seen in the dark field microscope. Okay, but in any of microscope also have a problem that we should encounter, and we also have a solution for it. And in this case for dark field microscope, the problem that we encounter when we using a dark field microscope, actually the first one is insufficient illumination to observe the specimen, or we can say the specimen are looking more faint, and this can be solved by raising the intensity of maximum or replace the light source. Okay, so in dark field microscope you can just maximize your light source to make your specimen to be visible, but bear in mind your specimen might not, some of your specimen might not like uh, a huge amount of light, so be careful when you maximize your light source in the dark field microscope. Okay, so considering your specimen. Okay, the second problem is the slide has a dark spot in the center of the field of the view, which is this problem can be solved by properly align and focusing the condenser will address this issue. Usually when you change the condenser are not in the proper position that you want when using a dark field illumination. So bear in mind when changing the condenser, check the position which is either is correct or not. Okay, so the third problem is the color appear in the background, or the background is unevenly lit with a gray cast in the dark field microscope. So usually this can be done or solved by remount the sample. Okay, I think that's a simple mistake, and we just need to remount the sample on the bright field, the dark field microscope. Next problem is the periphery of the view field is bright on one side only, which is the dark field is not fully darkened uh, background. So you should check the nose piece to ensure the objective lens are in the correctly position or is symmetrically on optical edges of the microscope, which is sometimes it's happen your objective lens are not aligned with your optical edges of the microscope. So carefully check, and usually you need to check either is exist or not with your microscope. And lastly of the problem that you probably encounter in the dark field microscope is the image of the specimen are not very clear and lacking insufficient of contrast and details, or I can say that your specimen are not fully recognized because in the dark field microscope, there is some specimen not might be suitable for the dark field microscopy. Okay, that's why we have many type of microscopy, which is that suitable and more specific to some of organisms. So in the dark field microscope, not all the microorganism can be seen under the dark field illumination. So bear in mind, there is only some of the example that can be see in the dark field microscope. Okay, and I think that's all from me and on my group about the dark field microscope. So thank you for your attention, and thank you for hearing our presentation.